thanks to all of you for joining today to learn more about the dissociation and isolation of solid tumor cells for optimized downstream analysis. So now you can see the agenda of this talk today. I want to start with the dissociation of solid tissues, as this is really one important prerequisite for all the things I will show you downstream of that. Then I want to focus on the isolation of tumor cells and subpopulations thereof, and that will be the major part of this webinar. And lastly, I will also discuss briefly the isolation of TIL. However, there will be also a colleague of mine giving a seminar in particular on this topic. So here you can see the cancer research workflow that we have developed over the past couple of years at Miltenyi. And you see it's really starting with the solid tissue where we have optimized and developed a max tissue storage solution, which I will not discuss today as this is already pretty well established in the market. However, I mean, the aim of this is really if you have your tissues and you cannot process them right away, but you have, for example, to store them overnight or over the weekend, or if you want to ship them to another site for processing, as we frequently do it actually in-house, then you can use this tissue storage solution to store your tissue prior to dissociation and downstream processing. Then the next step is, of course, the sample preparation. So getting a single cell suspension from your solid tumor tissue or any other kind of tissue. And for this, I will discuss mostly the tumor dissociation kits today. And then following this, when you have your viable single cell suspension, you can go on for cell isolation to really isolate the target cells that you're interested in, to then go on for any kind of further downstream analysis, molecular, culture, whatever you want to do. So let's start with sample preparation aspect. And you know that solid tissues are built up of a mixture of cells that are linked by cell-cell adhesion molecules, extracellular matrix, basal membranes. So you have two principal goals. The first one is, of course, very obvious. So you want to disrupt the extracellular matrix and cell adhesion components without destroying the integrity of the cell membrane. And for this, usually enzymatic digestion is the way to go. However, there's also a second goal that is not so obvious and not considered during many dissociation procedures. And that is that you might want to preserve your cell surface epitopes. For example, if you want to isolate your cell type of choice based on cell surface markers, you want to do flow analysis. But even if you want to do functional analysis and you need the cell surface receptors, such as the growth factor receptors, intact, then of course mechanical disruption is the way to go. However, a combination of both mechanical and enzymatic treatment is usually optimal for efficient dissociation. And that is what we have really optimized for a couple of years to bring out the optimal enzyme combinations, as shown here, the Tumor Dissociation Kit, which is composed of a mixture of 3 highly purified enzymes to reduce the lot-to-lot variation that you can get if you use, for example, bulky enzymes that still have tryptic, chemotryptic background activities like a lot of collagenases actually have. And this we combine with optimized and user-independent mechanical disruption by our GentleMax instrument in combination with our tube-based system. And actually, to learn a little bit more about your application, we would like to answer you a poll that will just come up right now. Okay, before I really go on to the details of tissue dissociation, I would also do a short excursus, let's say, and tell you a little bit about the tips and tricks that we have learned over the last couple of years when working with dissociated solid tissues, in particular tumor tissues, because they are actually quite different to hematopoietic tissues like PBMC or bone marrow. And you will see a lot of dot plots during my presentation, so I want to make sure that you really can follow what I'm using them for and what are the particular things I want to point out. And on this slide, you see that in contrast to many other like cell lines or PBMC, there's a significant heterogeneity in dissociated tumors. So here you see 3 different examples of a dissociated tumor tissue, and you see they also look very, very different among each other. And this, of course, depends on the tumor type, but also varies from patient to patient. And therefore, what we are using is usually a lot of backgating, and this you can see here on this slide. So we identify the major lineages such as the red blood cells, the white blood cells by known markers, and then we gate back these markers as shown here in red and blue, for example, and in green for the tumor cells. And on the right-hand side, you see the forward side scatter gates, and then you see where these populations are actually located in a dot plot. And then you can draw the gate, the initial gate, in a forward side scatter dot plot based on the red blood cells, because they will be the smallest cells, and then therefore you are not at risk of cutting out any relevant cell type. However, what you also see is that depending on the two different tumors that you see here, the upper and the lower panel, the composition is very different. So when you try to gate out red blood cells just by forward side scatter gating, you might lose red blood. You might lose also other nucleated cells. So therefore, usually we don't do that. We just gate out debris, and then we gate out red blood cells by cell surface markers. As you can see here, in human system, that would be glycophorin A. In the mouse system, it would be TER119. And then, as you see here in the lower left panel, you can also combine this with propylium iodide for cell viability, gate the dead cells and the red blood cells out, and then you can get an accurate counting of your nucleated cells and nucleated cell subpopulations. So another question that we are frequently asked is why we use a logarithmic scale in the forward side scatter dot plots. And that is pretty specific to solid tissues, I would say. And here's the reason why. As you can see in the upper left dot plot, here we dissociated a xenograft, and we stained the human EpCAM positive cells and the mouse, here anti-mouse fits positive cells, and then gated them back in blue and in red into the forward side scatter gate. So you see the red arrow marks the small mouse cells, the blue arrow marks the large tumor cells. And then in the lower panel you see in the hyperlook, that looks pretty nice. It's a nice resolution. You can see all these populations. If you now transform that to a logarithmic 3 scale, then you already see, okay, that's getting pretty broad, and you can still resolve the tumor cells, the human tumor cells and the mouse cells, but the debris is already on the axis, on the sum channel. And then if you even go to the linear scale, which most people use when using hematopoietic tissues, you see you can now resolve the debris, lower left, and then the red mouse cells, but the human tumor cells, they are so different in size that they are already on the sum channels on the axis on the right and then on the upper part. Of course, you can also decrease the detector laser power to bring the tumor cells back into the gate, but then you will not be able to distinguish between the mouse cells and the debris. So we really prefer the hyperlogarithmic scale in order to get a good resolution of these very heterogeneous tissues. And last aspect of particular pitfalls when working with dissociated tumor samples is that tumor cells after dissociation can be very sticky. And that is a problem, of course, if you work with live dead cell dyes, such as propylium iodide. So you see here in the unstained upper left-hand sample, everything is okay. Then you set the gate. You stain that with propylium iodide to label dead cells. It should only be included in dead cells, but actually you see, oh wow, only 30% viability, so that looks pretty bad. If you combine that with an antibody such as CD24PE, which not labels to tumor cells, you still see about 30% viability. However, now if you stain the tumor cells by a PE antibody, like EpCAM PE in the lower right-hand side, you clearly see that you have two populations of viable, or three populations of viable, but also two populations of dead cells. The left population in the upper gate, you see these are the dead non-tumor cells, and the really lower left population, these are the viable non-tumor cells. But you see in EpCAM labeled, the big round population, these are actually the viable tumor cells, and above there's a smaller additional population, and now you see these are the dead tumor cells. But if you would not use both dimensions here, but just the one, let's say the diagonal, you could never see where you should set the gate. So just to keep it in mind, be careful when you exclude viable and dead cells when working with dissociated samples. So all this knowledge, of course, we apply then in analyzing and optimizing our dissociation procedures in order to, of course, get a good viability and a good yield after dissociation. But also we do a lot of functional analysis. We do a cell culture of the cells, as you see here in the bottom panel. For example, this is a human melanoma cultured after 2 days, and then after 10 days, where nicely the melanoma colonies are growing on a layer of fibroblasts, as it would be expected. So that's all concerning the first question that you have. Concerning the second one, the epitope preservation, we really spend a lot of time to analyze that, and this is shown here how we do it. We take PBMCs, so other blood cells and cell lines, and then we label them with cell surface markers and treat them with the enzymes that we use for dissociation. And then we check the expression level, so in flow cytometry it's the mean fluorescence intensity, to actually check if the expression level of this marker is altered by the enzymes that we use, which is not the case for these 3 markers, but of course there are many more markers, and that's why we do whole cell surface marker screenings, and we check all of these markers in the portfolio more or less. And the idea behind that is really that the researchers doing an experiment, planning an experiment, and designing their panels can look beforehand if there are markers, their cell surface epitopes will be affected by the enzymes, and not learn it the hard way by just seeing, oh, my cell population is gone, I cannot detect it anymore. So you can just download these lists from our webpage, and then you can design your panels based on this. And as you see, only about 5% of the markers are actually affected by our enzyme composition. When you compare this to trypsin or other very aggressive enzymes like dispase or papain, usually more than 50% of the epitopes are affected. And because we use this mixture of 3 highly purified enzymes, but they are solely packed, you can see by reducing enzyme R, almost all of these markers can be preserved. And this only you buy in, of course, by a little bit lower yield and viability of the very tightly connected cells like the tumor cells and the endothelial cells, which are closely connected. But if you're analyzing immune markers, that might be the method of choice actually. So this is also seen, of course, in a lot of other applications, and this is, for example, from one of our collaboration partners, David Gilham, who was at that time based in Manchester, working on tumor-infiltrating lymphocytes from renal cell carcinoma. And what you can see here on the left-hand side is the comparison of our GentleMax Tumor Dissociation Kit digestion as compared to the published overnight digestion, only with collagenase. But again, as I mentioned, collagenases frequently have unspecific tryptic and chemotryptic activities. You see during this incubation with the overnight digest, you lose the CD4 and CD8 signal, while you can nicely depict the populations in our dissociation kit. And moreover, also because of the very gentle dissociation procedure, you see that the functionality of the cells is actually significantly improved. So by co-culturing the isolated TILs with autologous tumor cells from these patients, you see that here by readout interferon gamma release by these co-cultures, the T-cell activation is much better with our procedure than with the classical overnight digest. Just as a side note, next to the human tumor dissociation kit, which is used for the dissociation of human tumors, so everything that comes directly out of a human, but also of xenografts, so a human tumor growing in a mouse, we also have a mouse tumor dissociation kit, which is optimized towards the dissociation of syngeneic, so grafted mouse tumors, but also genetically induced mouse tumors. And basically the same applies here. And also for this, we have an epitope preservation list, of course, then for the marine epitopes. So if you're working with syngeneic mouse models, for example, in immuno-oncology, you can design your panels based on these stability data. So the last aspect concerning the dissociation of this viable tissue is actually pretty surprising, because there are always some approaches to do tissue dissociation purely on a mechanical basis in order to avoid any epitope stability issues. So meaning that when you do not use enzymes, the enzymes cannot harm your epitopes. However, what we figured out, actually a colleague of mine working with TILs for many years now, it is possible to dissociate some tumors, so the very soft ones like B16 mouse melanomas, without enzymes. And you, of course, get a lower yield and viability of the tumor cells, the endothelial cells, so again, a very tightly connected tissue cells. However, the CD45, so the overall TIL counts look pretty normal. However, if you now look really at the very specific T cells, such as the TIM3 positive, PD1 positive, LAG3 positive, tumor reactive T cells, which are tightly connected to the tumor cells, you see with our GentleMax procedure and the tumor dissociation kit, you get a very nice population in the upper panel. If you use a pure mechanical dissociation, you completely lose this population, not because of epitope preservation issues, but just because these cells are lost because of the mechanical stress. So in just a small side note, this was all now dissociation of viable tissues. We now have also recently released a method for dissociation and getting a single cell suspension out of formalin-fixed paraffin-embedded tissues, which you then can use for flow sorting, for example, for isolation of the tumor cells, flow analysis, and also DNA downstream analysis. And my colleague Bastian will also give a webinar beginning of June on exactly this tissue. So if you're working with FFPE tissue, please join the webinar and learn more about this very novel application. So with this, I want to finish with the sample preparation part and now go on for the cell isolation. Now you have your viable single cell suspension in a good shape, and now you can isolate your target cells. And of course at Miltenyi, we mostly focus on magnetic cell separation. Most of you might know the system, however, I'll briefly explain it. The principle is rather easy. So you start with a heterogeneous cell composition showing in the left-hand side, and then we use antibodies that are specific towards the epitope of your target cell that are coupled to paramagnetic nanoparticles, very small ones, like 50 nanometer. That's the size of a virus, and therefore also guarantees that the cells are not affected by this. And then we run it over a column that is placed in a magnetic field. The non-labeled cells are just in a flow-through fraction. If you remove the column from the magnetic field, you can elute your target cells. And of course, this is also possible vice versa. If you do not know an epitope of your target cell, or if you do not want to touch your target cell with an antibody, you can also label the non-target cells, and they will stay in the column, and your target cells then, of course, will be in the flow-through fraction. And the methods I will show you right now are all based actually on these untouched, or we call it negative selection approaches. So let's talk about now the isolation of untouched tumor cells. In principle, there are three major starting materials where you can isolate tumor cells from. Xenografted tumors, so human tumors growing in a mouse, pure mouse tumors growing in a mouse setting, and of course primary human tumors. And for all of these tumors, we have developed optimized methods for dissociation and also for isolation of the cells, which I will show you now. I will go more into detail for the xenografted tumors now, because the principle and the method are very comparable among all of these. So let's focus on a xenografted tumor. First of all, we have to ask ourselves, why should I deplete the mouse cells out of a xenografted tumor? Is it important or not? And of course, it always depends on the downstream application, but what you can see here is three different models of non-small cell lung cancer xenografts in mice. And you see the degree of mouse cell infiltration is very different among the different patient tumors. However, when you graft two tumors in each flank, or in both flanks of the mice, you see that even these two tumors, which are coming from the same patient tumor, have a very heterogeneous degree of infiltration. So if you would do, for example, a molecular analysis out of this, you would have a very influential bias towards the number of infiltrating mouse cells for your downstream analysis, cell culture, molecular analysis, Western blotting, everything you want to normalize your target cells towards the starting material. And for this, we have come up with a solution some years ago by doing large cell surface marker screening. So we tested a couple of hundred antibodies on all different mouse strains, all different mouse tissues, and figured out a combination of now actually 5 antibodies that label all of the mouse cells from all the different organs, from all the different mouse strains. So after you're having dissociated your tissue, you have your single cell suspension, you just add this cocktail of 5 antibodies that are coupled to magnetic particles, incubate it for 15 minutes, you run it over a column, and your human tumor cells are flowing through the column. If you want, you can then remove the column and elute your mouse cells. But as this is a negative approach, it works for any kind of human tumor cell or even non-tumor cell. So any kind of human cell that you graft into a mouse can be isolated by this procedure, and the whole procedure just takes about 20 minutes. And in the lower panel, you can see the example. So you have the EpCAM-positive human tumor cells and the EpCAM-negative mouse cells. And then after 20 minutes, you have a pure fraction, in a negative fraction of tumor cells. And this is, as I said, independent on any kind of positive marker. So it works for any kind of solid tumors, like here the renal, lung, and bladder cancer xenografts, but it also works for leukemias, lymphomas, and also even non-tumor cells, as I will also show you in a couple of minutes. Of course, it makes a big difference in culture, and frequently xenografts are actually used to expand the human tumor material in order then to dissociate it and to generate cell cultures for drug testing. And if you just dissociate your xenograft and you put it in a culture plate, it looks like the panel I'm showing you right now. So you have vimentin-positive mouse fibroblasts, and you have, for example, if you are using a carcinoma, EpCAM-positive human tumor cells. However, if you now do a drug toxicity assay on this here, it would be very hard to distinguish which effects come from your target cell and which from your contaminating cell fractions. By using just this 20-minute mouse cell removal fraction method, you then have your isolated tumor cells and there are barely any mouse cells left in the fraction. And now, of course, you can do a very homogeneous readout. And what was pretty surprising to us also is next to removing all the mouse cells, one of our initial test sites actually figured out that you also remove the vast majority of the dead cells in the debris. And this is data from Jeremy Rich from the Cleveland Clinic. His group is working on human glioblastoma cells, which they tag with GFP, and then autotopically transplant them into mouse brains. And if you transplant that into a mouse brain, and if you then dissociate an adult mouse brain, most of the things that or events you will have later on are actually debris and dead cells because of all the myelin that you destroy during the dissociation. So you see in A that the cells only make a minor fraction after dissociation. And then, of course, in B, you see the human tumor cell content. And now what you see is that next to isolating or depleting all the mouse cells is also in C that you have really cleaned up your samples. So you have a significantly lower amount of dead cells and debris. And this is very surprising and, of course, very beneficial for a lot of downstream applications, including, for example, next-generation sequencing, as you see here, because we want to evaluate the debris removal step also on this in this respect, and did it in collaboration with Charles River on 3 different kind of xenografts, lung, kidney, and bladder cancer xenografts. And what you can see here is really that you get a significant improvement of your sequencing results, so higher cluster density, higher yield, and therefore also higher read counts. So you really improve that data because you start with a really higher quality sample in the beginning. And next to, of course, doing that on pure, let's say, tumor tissue, you can also look for circulating tumor cells or disseminated tumor cells. And this is just one example data of the Massimo Moro lab from Italy, which are looking into disseminated tumor cells. So they set a xenograft in an animal, and then after a couple of weeks, they look for human disseminated cells into the lung for metastatic spread. And if you then take out the lung and you dissociate it, usually the human events are so rare that it's really below the detection level of flow cytometry to enumerate them. However, if you now do the mouse cell depletion before, then you can significantly enrich it by several log decades, as you see here in this example, one human cell in almost 340,000 mouse cells, whereas after the depletion, one human cell in 400 mouse cells, which is now at a level where you can really enumerate it by flow cytometry. And the same holds true for human circulating tumor cells in xenografts, of course. So last example for the mouse cell depletion kit is what I mentioned in the beginning, that it's not only working for tumor tissue, but also for non-tumor tissue because it's marker independent. And this is data from John Dick's lab in Toronto, Canada, which are using this kit actually for humanized mice. So they do a human hematopoietic stem cell transplantation, either from umbilical cord blood or from ALL bone marrow. And then, so they reconstitute the bone marrow of the mice with a human immune system. And then after a couple of weeks, of course, you have a very heterogeneous chimerism grafting. And then they want to homogenize the samples before they go on with the downstream analysis. And this is what you see in the right here. The black bars are just before the depletion, very heterogeneous sample composition. And then after mouse cell depletion, you nicely normalize all the samples and then can go on and reduce this, let's say, cause of irreproducibility just before your downstream analysis. So I could not go into further molecular analysis due to time reasons, but if you're interested in that, we have some scientific posters available, which you can download from the webpage or just drop us an email, and then we can send you the posters, in particular on the NGS aspect of mouse cell depletion. So next, I want to discuss the isolation of mouse tumor cells from primary mouse tumors. And the principle, as I mentioned, is completely the same. However, here the challenge is more that it's, of course, harder to find suitable markers because here you cannot use species-specific antibodies. You have your target cells are mouse and also your contaminating cells are mouse. But again, we were able to identify a marker composition in a large cell surface marker screening to now efficiently isolate also primary or genetically induced mouse tumor cells. And this is shown here for 3 frequently used models, the CD26, B16, and 41 tumors. And we have tagged these tumor cells with GFP before transplantation into the animals to really track them. And what you can see here, again, it's a depletion approach, takes only 20 minutes, and you end up with very pure isolated tumor cells. And the same holds true, of course, in the culture. When you plate the original fraction, you see a mixture of GFP-positive tumor cells and smooth muscle-lectin expressing activated fibroblasts. After dissociation and after isolation, you see very nice and pure cultures of the isolated tumor cells. The last aspect here is, of this part is the isolation of human tumor cells straight out of patient material. And again, I have to say the principle is the same. It's a 20-minute procedure that is based just on the depletion of your unwanted cells. It's completely independent on your markers. And what you can see here, this is an original fraction from a breast cancer patient. You see the forward side scatter, again, very high heterogeneity. You see a lot of white blood cells labeled by CD45, red blood cells by glycophorin A, endothelial cells CD31 positive, fibroblasts, and also the EpCAM positive tumor cells. You apply the labeling, you run it over column, 20 minutes later, you have a pure human tumor cell fraction. And again, if you compare the culture of these cells, you see if you plate the original fraction, you see compared to the other starting materials, you see tumor cell colonies stained in EpCAM and a lot of fibroblasts that are also giving you a high risk of losing this culture because the fibroblasts, they plate efficiently, they grow very fast, and frequently you lose a culture by overgrowing fibroblasts. Whereas when you plate the cells after isolation, you will have a very nice and homogeneous culture, and you completely avoid the risk of losing your culture by overgrowing fibroblasts. And of course, also if you want to do drug toxicity, again, that will be a much more homogeneous starting population. What is very important is that it does not only work with, let's say, easy samples where you already have a high initial content of tumor cells, but it also works with challenging samples such as pleural effusion or ascites. And what you can see here, this is a pleural effusion from an ovarian carcinoma patient. And you see the tumor content, as labeled here by EpCAM staining, is very low. It's usually below 1% in pleural effusions. But again, applying the same procedure, 20 minutes after, you have a very pure population of your tumor cells that you can now go into culture or do molecular analysis or whatever you want to do. And this is from the very same patient. We plated the original fraction, let it adhere and grow for 3 days. And then we washed it several times with PBS, and you see in the contrast, okay, there are some tumor cells in there, but there are also a lot of other cells and debris, fibroblasts, blood cells. Whereas when you just isolate the cells and plate them, very same patient, but this time without washing with PBS, you see a very nice and homogeneous tumor cell cultures. You see a lot of multinucleated cells, which is a hallmark of ovarian carcinoma. And of course, you can then go on and expand these cells for further analysis. So we were wondering, what is the influence of the isolation of the tumor cells on next generation sequencing? Because this is one of the major applications that people are doing right now. So what we have done is we received tumors, we dissociated them based on a Tumor Dissociation Kit, isolated tumor cells and also the stroma cells, and then we did exome sequencing on the Illumina platform on the isolated tumor cells and also on the bulk tumor, which is currently the most frequently used method. So you just take a chunk of tumor, you lyse it, isolate DNA or RNA, and then you do the sequencing. And what you can see here is the loss of heterozygosity from 2 different ovarian carcinomas, one ovarian carcinoma metastasis and also one tumoma. And what you can see is that you have a much more accurate detection of the loss of heterozygosity events in the isolated tumor cells, because you do not have the contaminating DNA from all the non-tumor cells, such as the white blood cells and the fibroblasts and endothelial cells in the bulk tumor samples. And this you can of course also see in the mutation calling. So this is a mutation in the P53 gene in this ovarian carcinoma patient, and also in the primary tumor and in the metastasis. And when you just sequence the bulk tumor, as shown here in blue, you see, okay, that looks like a heterozygous loss of function in P53, but as we know from the flow analysis, there was about 50% tumor cell content in these samples. And we know, okay, actually that's coming from the impurity of the sample. So after isolating the tumor cells, you actually see it's a homozygous loss of function mutation in this P53. And this, of course, is not so challenging because the tumor cell content was high and we are looking for a clonal mutation. But if you now look at low tumor cell content and subclonal mutations, you're really going into the detection limit. And this is shown here when we isolate the tumoma, which is usually a tumoma of the carcinoma of the thymus, where after dissociation you have usually less than 1 or 2% tumor cells in your fraction. And if you sequence these, you see here on panel A, this is a proto-oncogenic gene, the beta-catenin 1 version. And when just sequencing the bulk tumor, you do not detect any mutation in the reads, as you can see here in the gray shaped area. Whereas when we isolate the tumor cells to about 90%, we actually see there's a heterozygous loss of function, a heterozygous gain of function in this tumor. And even more prominent, I think, is the example here in B, where by sequencing the bulk tumor, we saw, okay, there's a heterozygous loss of function in this tumor suppressor, the check gene, but we knew that there was only less than 2% of tumor cells in the initial fraction. Whereas when we isolated the tumor cells, as shown here in orange, we see, oh, actually it's a homozygous loss of function, meaning that actually this patient had a germline mutation already, a heterozygous germline mutation in this gene, and during tumorigenesis, this was a loss of heterozygosity event, turning it into a homozygous loss of function, and was probably one of the initiating events during tumor formation. So very briefly, I want also to touch on RNA analysis, because this was DNA analysis, but of course you think RNA acts in another level of complexity, because here you not only have two information points per cell, like in a DNA based on the chromosomes, but all the different RNA species will have very different expression levels. So there it's much harder to really dissect bias from the real events. So we have just concluded a small study where we have isolated a tumor subpopulation, CD133 positive tumor cells from ovarian carcinoma. And usually what is mostly published and what most people are doing, they take the bulk tumor, dissociate it, and straight go on for isolation of their CD133 positive tumor subpopulation or CD44 positive, 24 negative population. Let's call it cancer stem cells or not, doesn't matter, it's just meaning you isolate a tumor cell subpopulation. Whereas we have compared this standard procedure to first isolating tumor cells and then isolating the tumor subpopulation out of these tumor cells, which is possible because it's an untouched separation, so you can do an additional magnetic separation afterwards again. And this, just briefly looking at it, shows you the results. And what you can see here is that when you compare the tumor subpopulation of CD133 positive cells to the bulk tumor, you see a lot of difference. But you see also the same difference when you just compare the isolated tumor cells to the bulk tumor, meaning that most of these signals that you get actually is just a difference that you removed all the stromal cells from your sample. And actually, if you look at C here, you see that really, if you want to challenge what are the differences among the tumor subpopulation and the other tumor cells, the differences are really minor. And if you now have also all the stromal cells in your control fraction, they will give you a very big expression difference, and therefore you would lose your relevant genes just in background noise, because all these highly expressed, for example, immune panel genes will just level out your real target genes. And again, due to time reasons, I will not go into the details here, but if you're interested in that and also comparing this method to laser microdissection, please be aware that there are several scientific posters that you can download from the webpage or get just from us directly. So now coming to the last topic, and this is the isolation of tumor-infiltrating blood cells or leukocytes from tumor tissue. And let's start with syngeneic mouse tumors because they are the most frequently used model system right now to analyze, for example, in immuno-oncology. And this now is an example for the positive selection. So what we are using is we use antibodies that are coupled to that are specific for CD45 and coupled to the magnetic beads to really allow for a pan white blood cell isolation straight from tumor tissue. And these reagents are now really optimized towards working with dissociated tumor samples because, I mean, there have been CD45 selection reagents available for years or even decades. However, they do not really work on tumor tissue. So now we've brought up this new product line of TIL-specific isolation reagents, and they are really optimized to work straight from dissociated tumor tissue. And as mentioned, I will just go onto touch the basis here, but there will be a dedicated webinar by my colleague Cesar on end of May on the optimized workflow for the isolation of tumor-infiltrating immune cells, leading to fast and improved flow analysis. However, let's briefly touch on this. And I would say the most frequently basic question is, why should I isolate my TILs? Because I just want to do flow analysis, so I can just gauge on them based on CD45, and then it's fine. Okay, let's just do a calculation based on a real-life example. When we work, for example, with B16 melanoma, we inject 500,000 melanoma cells subcutaneously. We wait 1 to 2 weeks, and then we have 200 to 500 milligram of tumor tissue, which will give you about 10 to 20 million cells. So if you now want to do flow analysis, let's say you will start with 10 million cells. B16 is not very well infiltrated, so you have about 2% CD45 positive cells. 10% of these 2% are CD3 positive T cells, and 16% out of the 10%, out of the 2% are CD8 positive cells. So even starting with 10 million cells, you will only have 3,200 CD8 positive T cells in this sample. And then if you want to now analyze a subpopulation of CD8 cells, such as an antigen-specific CD8 T cell, which is about 5% of this population, if you just want to acquire 100 events of your target population, you need to acquire more than 6 million total cells from your sample. If you run this flow cytometer at 4,000 events per second, it will take you 26 minutes. And if you have 9 samples, which is really not a lot, so 3 groups and 3 replicates, it will take you 4 hours in front of the flow cytometer. And a lot of reagents, because you have to stain really high numbers of cells. If you do the TIL selection before, to acquire 100 events of your target population, you would just have to acquire a little bit more than 100,000 of the total events after selection. That will take you 33 seconds. And for 9 samples, it will take you 5 minutes. Of course, then plus the 20 minutes for the separation. But you see very fast, if you have more samples, which most of the people have, it makes a huge difference. And we have optimized this TIL procedure to really work with all the kind of different model systems, CD26, B1641, but also other genetically induced and other activated tumors. And all of them give you a pretty nice and reproducible result, as you can also see here. Of course, the final purity is determined a little bit by the initial frequency, but still the factor of enrichment is always very significant. And usually the more significant, the lower the initial frequency is. This is just one example. What I have just pointed out in a numbers game is just we said, okay, we will not acquire more samples, but we just do a flow analysis on a B16 tumor without isolation, what you see here, and we just acquire 100,000 events to keep the time the same for the analysis. So this is the information on the subpopulations that you get when you acquire 100,000 events on the immune subpopulations. When you do the isolation, you see you start with a much more pure population in the beginning, and then with acquiring the same number of 100,000 events, you can even go down to small subpopulation like dendritic cells, tumor-associated macrophages, and can even do a sub-phenotyping of these without using more time and more effort at the flow cytometer. And this is also now the time where we want to ask you the second poll, and so please give us an answer to this. Thank you. Okay, thanks for answering the poll. And so now I just want to give a very brief overview, because a lot of people working with syngeneic mouse models actually do not have 5 or 10 samples, but they really work with large cohort sizes, such as 24, 48, and even more animals at a time. And therefore, we have spent some time in really parallelizing and also automating this workflow. And here you can see the manual separation, where it is easily doable of doing 8 separations in parallel with a stand and a magnet. However, we've also optimized this method towards the multi-mark cell 24, so that you can easily do 24 separations in parallel. And if you even have a higher demand for automation, we've now also included the multi-mark cell 24 plus in a liquid handling robot station, so that you can just insert your first 24 samples, you press start, the instrument will do all the separation for you, you go away, and then you come back with the next 24 samples once this instrument is finished. So next to the optimization of this protocol for mouse TIL isolation, we have now also recently released the human counterpart, which now also shows you very nice results when working with primary human material. As you see here, this is a human colon carcinoma, which we have dissociated. And then you see again in the flow-through fraction, you have your CD45 negative, mostly tumor cells, endothelial cells, fibroblasts. And then in the positive fraction, your isolated CD45 positive cells. And this, of course, because it's human-specific, not only works with the primary human material, but also with humanized animals, which are frequently used now in immuno-oncology applications. So, and this is data from one of our test sites at Pfizer, where they use mice carrying a human tumor and also a humanized immune system, and then they are testing, for example, bispecific antibodies, T-cell engagers, and then want to isolate their T cells and that are actually infiltrated in a human tumor, in the human tumor xenograft, for downstream analysis. And this, you can see, also nicely works with this CD45 TIL microbeads human, because you will just isolate the human CD45 cells out of the tumor and will not get any contamination by the mouse cells because of the lineage-specific antibody. And now it's time for our last poll, so please, we would like to answer you. Okay, in the last, let's say, couple of seconds, I would like just give you one note, because you have seen a lot of flow analysis that I have showed you in this webinar. And you might be aware of this big debate about the reproducibility crisis in research. And that's actually a major part of this is coming from non-validated antibodies. And it's pretty funny, because at Milton, even before that debate, so many years back, we've decided to turn our whole antibody portfolio into recombinantly engineered antibodies already, because that will give you very reproducible standardized results, because the antibodies are all sequence validated. You only have one heavy chain, one light chain, and because they are recombinantly engineered and produced, you always have the same. There's no lot-to-lot variability. In addition, we engineered the constant region so that there's no background Fc gamma receptor binding anymore. And in addition, also the constant region is only one isotype. All the reaffinity antibodies are human IgG1, which of course simplifies your flow analysis, because for your whole reaffinity panel, you just need one isotype control anymore. And just to show you the advantage of these antibodies, when working here with splenocytes or tumor cells, you see this is a CD8 staining. And then you compare the reaffinity antibody to a classical hybridoma antibody, you see in the spleen that all looks pretty good. The antibody does not give you a lot of background. However, in dissociated tumors, we know that a lot of cells upregulate the Fc gamma receptors. And what you can see here, the reaffinity antibody is not affected by this. The hybridoma antibody, as you can see in the bottom panel, gives you a high background when working with dissociated tumor cells. Of course, with FCR blocking, you can diminish this background, but you still see about a 5% background, and you see a pretty broad negative population. Whereas the reaffinity antibody, no matter if you use FCR blocking or not, will always give you a very nice and clean staining. So with this, I want to sum up, and I hope I could convince you today that optimized and automated tissue dissociation really improves your downstream processing and analysis of target cell fractions. that the MAX technology allows for the fast and reliable isolation of target cells from various lineages, and that generic cell type specific and also combination sorting strategies enable sorting of all major tumor subpopulations. So with this, I want to thank you very much for your attention. I want to, of course, thank my team because they are doing all the work. I'm just presenting results, but they are coming from the lab, and these are the people that really stay in the lab and generate the results. I want to thank my collaboration partners and also a lot of additional Miltenyi colleagues. So thanks all of you. And now I would be happy to take some questions.